All right, let's jump right in. Today we're looking at California Encephalitis Virus, but not just as another name on a long list of pathogens. We're treating it like a case file, a genuine diagnostic puzzle for clinicians and, for us, in the microbiology lab.
You know the drill, right? A patient comes in with a serious neuroinvasive disease, we run all the standard panels, and more often than not, we get nothing back. It's a huge question mark. And this isn't some minor frustration, it's a massive gap in our diagnostic world.
And this really puts the scale of that problem into perspective. The landmark California Encephalitis Project found that even after throwing everything they had at it, the cause for a staggering 62% of encephalitis cases was a total mystery. So the question is, what are we all missing?
Well, this explainer is all about a potential hidden culprit. We're going to explore whether a virus like California encephalitis is truly rare or if it's just an artifact of us not having the right tools to find it. Just so you know, all the information we're covering was pulled together using a Gemini deep search, and you can dig into all the sources yourself in the linked NotebookLM document.
Okay, so first up, framing the virus. Let's look at it as an unexplained neurological threat, especially one we might see in a traveler.
So what exactly are we dealing with here? It's a neurotropic arbovirus, which means it loves nerve tissue and gets around via arthropods, specifically Aedes mosquitoes. Now, for us here in the Netherlands, this is really important. It's an imported pathogen. You're not going to see this unless your patient has a travel history to North America. And that last point is the real kicker. Its rarity might be a total misnomer, just a reflection of the diagnostic tools we've been using up until now.
To really understand how to hunt this thing down, we've got to know what it looks like. So let's build a proper profile of our suspect. And here's our first piece of solid intel. This chart places it right where it belongs. It's an enveloped, negative-sense RNA virus, part of the Bunyaviridae family. And look, that's not just trivia for an exam. That classification tells us a ton about its structure, how it replicates, and where we should be aiming our diagnostic tools.
And here's the full family tree. This level of detail is absolutely critical for us in the lab. When you're designing primers for a PCR or trying to make sense of sequencing data, knowing you're looking for an orthobunyovirus is what gives you that specificity and helps you avoid confusing it with something else.
Taking a closer look, it's pretty much a classic design. You've got a spherical, enveloped particle about 90 to 100 nanometers across. Those little spikes on the surface? Those are the GN and GC glycoproteins. They're like the keys the virus uses to unlock and enter host cells. But the really interesting part is inside. Its genome is split into 3 separate segments of negative-sense RNA.
And that is why the segmented genome is such a big deal from a public health standpoint. It's the exact same trick that influenza pulls. If 2 different strains of the virus infect the same cell, they can literally shuffle their genetic cards and create a brand new hybrid virus. This reassortment means we're constantly facing the potential for new and maybe more dangerous strains to just pop into existence.
Okay, so we've got a profile of the virus. Now let's see what it actually does when it gets into a patient. What does the disease look like?
If you remember one thing about the clinical side of this, make it this. Age is everything. The patient's age is the single biggest factor that determines how severe this infection is going to be.
And the difference is just night and day. In adults, an infection is usually no big deal, maybe even asymptomatic. But in kids, it's a completely different story. It can be a devastating neuroinvasive disease, causing encephalitis, seizures in about half of the cases, and for up to 20% of those kids who survive, there's a real risk of long-term neurological problems.
So, knowing what this looks like in the clinic, how do we actually nail the diagnosis in the lab? This gets us right to the heart of the challenge and the modern tools that are, well, frankly, changing the entire game.
The journey here is really incredible. We started with viral culture. It's slow, takes weeks, and you need a BSL-3 lab, but we still use it to confirm we have live infectious virus. You grow it in vero cells and watch for those classic cytopathic effects, the cells rounding up and detaching. Then came RT-PCR, which was a revolution. Fast, sensitive, and our go-to for an acute diagnosis. But now we have metagenomic next-generation sequencing, or mNGS, and it's a whole new way of thinking.
And this right here is the paradigm shift. RT-PCR is hypothesis-driven. You have to suspect the virus to even run the test. It answers a very specific question. Is this virus here? But mNGS, it's completely hypothesis-free. It answers a much bigger, much more powerful question. What microbes are in this sample? Period. And that is how we can start chipping away at that 62% of unknown cases.
Okay, so let's say we've used these new tools and we found it. We've got a diagnosis. What happens next? Let's talk about management and, maybe even more importantly, prevention.
Let's just be blunt about this. There is no specific effective antiviral drug for California encephalitis virus. And that stark reality makes a fast, accurate diagnosis even more vital. Not to pick a drug, but to guide supportive care and, crucially, to rule out all the other causes of encephalitis that we can treat.
Now, for our daily practice here in the Netherlands, this concept is so important. We operate on a guideline by proxy. You can search all you want, you won't find a specific RIVM guideline for California encephalitis virus. Instead, what we do is manage the patient following our really solid national protocols for other severe viral CNS infections, like tick-borne encephalitis. This approach is totally in line with what international bodies like the WHO and CDC recommend, which is just intensive, high-quality supportive care.
And in kids, that supportive care has some really specific, non-negotiable points. Getting seizures under control is priority number 1, and the warning to avoid aspirin is absolute. You do not want to risk Reye syndrome. And so importantly, the job isn't done when the child leaves the hospital. We have to be planning for long-term, multidisciplinary follow-up right from the start, because that risk of lasting neurological damage is just too high.
So if there aren't any antiviral drugs, what do we even mean by resistance? Well, we have to reframe the question. It's about the virus's own built-in resistance to our immune system. Its main weapon is a nasty little thing called the NSS protein, and its job is to shut down our interferon response, basically cutting the wires on our body's alarm system. Because of this, we don't do clinical antiviral susceptibility testing. Sure, in a research setting, you might see a plaque reduction assay, but for now, that's purely academic.
Since treatment isn't an option, prevention becomes everything. And the strategy is pretty simple. Just don't get bitten by mosquitoes. That means using proper repellents like DEET, wearing long sleeves and pants, making sure your window and door screens are in good shape, and getting rid of any standing water where those Aedes mosquitoes love to breed. This is Public Health 101.
Which brings us right back to where we started. This quote just nails it, doesn't it? As we get better and better with unbiased tools like mNGS, we have to start questioning what we mean by a rare disease. We might just find out that California encephalitis virus and a lot of others like it aren't nearly as rare as we thought. We just weren't seeing them.
And that's our deep dive into the diagnostic challenge of California encephalitis virus. If this was helpful for your work, definitely subscribe for more briefings like this one. We'd really love to hear your thoughts, your own experiences, or any questions you have, so please drop them in the comments below. Thanks for tuning in.